In this chapter, we will explore the phases and enzymes involved in translation, a fundamental process in gene expression. You will also learn to contrast the features of prokaryotic and eukaryotic translation, including differences in timing, cellular location, and the enzymes involved. By the end of this lesson, you should be able to identify key translation components and appreciate the complexity of protein synthesis in both domains of life. The ribosome is a molecular machine responsible for synthesizing proteins. In bacteria, ribosomes are 70S in size and are composed of a large subunit containing 23S and 5S ribosomal RNAs, and a small subunit with 16S RNA. In contrast, eukaryotic ribosomes are 80S, composed of a large subunit with 23S, 5S, and 5.8 srRNAs, and a small subunit with 18 sRNA. Ribosomes can be free-floating in the cytoplasm or attached to the endoplasmic reticulum, which gives rise to the characteristic appearance of rough ore. On this slide, we visualize the structural differences between prokaryotic and eukaryotic ribosomes. Figure 16.01 displays the components of the bacterial ribosome, while Figure 16.02 presents the architecture of the eukaryotic 80S ribosome. These structural differences underlie key functional distinctions in how translation proceeds in each type of cell. Transfer RNA, or tRNA, is the adapter molecule that links mRNA codons to their corresponding amino acids during translation. A charged tRNA is one that has an amino acid loaded onto it. This charging is catalyzed by enzymes called aminoacyl-tRNA synthetases. tRNA plays a crucial bridging role, interpreting the genetic code and ensuring accurate protein synthesis. The adapter hypothesis, proposed by Francis Crick in 1955, suggested that free amino acids cannot align directly with mRNA codons. Instead, an adapter, tRNA, is necessary. Crick predicted there must be at least 20 different adapters, one for each amino acid, and that each amino acid must be loaded onto the correct tRNA. This concept laid the groundwork for understanding how tRNA reads the genetic code. All tRNAs share a common structural feature at their three ends: the sequence sequence, which is the attachment site for amino acids. Interestingly, most eukaryotic tRNA genes do not code for this sequence. Instead, it is added post-transcriptionally by tRNA nucleotidyl transferase. The amino acid attaches via an ester bond to either the 2 or 3 hydroxyl group of the terminal adenosine. Importantly, each tRNA is highly specific for its corresponding amino acid. After transcription, tRNA molecules undergo extensive modifications to enhance their function. These include methylation, reduction of double bonds, modification of pyrimidine rings, replacement of oxygen atoms with sulfur, and addition of large chemical groups. These modifications help stabilize tRNA structure and facilitate precise base pairing during translation. The secondary structure of tRNA resembles a 3-leaf clover, consisting of 5 major arms. These are the acceptor stem for amino acid attachment, the anticodon arm, which pairs with mRNA codons, the D arm, named for dihydrouridine, the TC arm, important for ribosome binding, and the variable arm, which varies in size and sequence. Figure 16.11 illustrates this highly conserved and functional structure. When folded into its 3-dimensional form, tRNA adopts an L-shaped tertiary structure, as shown in Figure 16.14. This configuration allows the tRNA to interact simultaneously with both the mRNA codon and the ribosome's catalytic site. It is this structure that enables the tRNA to deliver amino acids accurately during translation. Initially, scientists believed there were only 20 amino acids used in protein synthesis. However, the discovery of selenocysteine and pyrrolysine added new complexity. Each of these amino acids has its own unique tRNA and aminoacyl-tRNA synthetase. These enzymes are critical for linking the correct amino acid to its corresponding tRNA, ensuring fidelity in protein synthesis. Protein synthesis relies on the genetic code carried by messenger RNA, or mRNA. Each group of 3 adjacent bases on mRNA forms a codon, which specifies a particular amino acid. This code is degenerate, meaning multiple codons can encode the same amino acid. Point mutations can alter the codon, resulting in missense mutations, where one amino acid is substituted for another, or nonsense mutations, where a stop codon is introduced. Frameshift mutations, caused by insertions or deletions, can alter the reading frame and severely disrupt protein function. Translation proceeds from the 5 to 3 direction, while the resulting protein is built from N-terminus to C-terminus. In prokaryotes, transcription and translation are coupled, allowing proteins to be synthesized even before mRNA synthesis is complete. This table summarizes the genetic code, highlighting all 64 codons and their corresponding amino acids. Notably, there are 3 stop codons, UAA, UAG, and UGA, which signal termination of translation. The start codon August, which codes for methionine, also sets the reading frame. This table illustrates the redundancy and specificity of the genetic code, forming the molecular basis of protein synthesis. The 3 termination codons, UAA, UAG, and UGA, are referred to as nonsense codons. Mutations that convert a sense codon into a stop codon are thus classified as nonsense mutations. Historically, researchers identified and named these mutations based on the stop codon produced: amber for UAG, ochre for UAA, and opal for UGA. These mutations can halt protein synthesis prematurely, often leading to loss of function. The genetic code follows 5 key principles. First, it is non-overlapping, meaning each nucleotide is read only once. Second, it is commaless, with no gaps between codons. Third, it is nearly universal, such that genes from one organism can often be expressed in another. Fourth, it is degenerate, with most amino acids being encoded by more than one codon. Finally, the code is unambiguous, ensuring that each codon maps to only one amino acid. These features ensure both flexibility and fidelity in gene expression. An important experiment demonstrated that the specificity of the genetic code lies in the tRNA, not the amino acid. In this study, cysteine attached to terminases was chemically converted into alanine. Remarkably, alanine was inserted at the positions normally occupied by cysteine, proving that the ribosome reads the anticodon of the tRNA, not the chemical identity of the amino acid. This reinforces the central role of tRNA as the true interpreter of the genetic code. The wobble hypothesis explains how a single tRNA can recognize multiple codons. Because codons and anticodons bind in an anti-parallel fashion, the first 2 bases of the codon form strict Watson-Crick base pairs with the anticodon. However, there is flexibility, or wobble, at the 3rd base of the codon, allowing for non-standard base pairing. This mechanism contributes to the degeneracy of the genetic code and allows cells to maintain fewer distinct tRNAs. The bacterial ribosome consists of a 30S small subunit and a 50S large subunit, each with distinct structures and functions. The ribosome is a dynamic and precise machine capable of translating 10 to 20 codons per second with an error rate of less than 0.03%. The small subunit is responsible for decoding mRNA by verifying proper codon-anticodon pairing, while the large subunit carries out peptidotransferase activity, forming peptide bonds between amino acids. Structural analysis of the 70S ribosome has revealed three distinct binding sites. The A site, which holds the incoming aminoacyl-tRNA. The P site, which holds the tRNA carrying the growing peptide chain. And the E site, where deacylated tRNAs exit. These sites coordinate the stepwise movement of tRNAs and growing polypeptides during translation. Translation is divided into 4 stages. First is initiation, where the ribosome assembles around the start codon with the help of initiation factors. Second is elongation, where amino acids are sequentially added to the growing chain as tRNAs pair with codons on the mRNA. Third is termination, where release factors recognize stop codons and release the newly made protein. Finally, in the recycling stage, ribosomal subunits are disassembled and reused for a new round of translation. During initiation, bacterial mRNAs are often polycistronic, meaning they contain multiple open reading frames, each with its own start codon. Translation can begin as soon as the 5' end of the mRNA emerges from RNA polymerase, allowing transcription and translation to occur simultaneously. The ribosome must locate each start codon independently, enabling synthesis of multiple proteins from a single mRNA strand. This slide offers a recap of translation, highlighting the major steps: initiation, elongation, termination, and recycling. It emphasizes the precise coordination of multiple molecular players, from tRNAs and ribosomes to initiation and elongation factors. Each stage is carefully regulated to ensure accurate and efficient protein synthesis.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine-tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
In bacteria, two different methionines and two different tRNAs are involved in translation. Regular methionine paired with ternamate is used during elongation, while informal methionine, or FMET, paired with ternafmet, is used exclusively for initiation. The formal group ensures that FMET is only incorporated at the amino terminus. After translation begins, the formal group is removed by peptide deformylase, preparing the protein for further processing. The bacterial 70S ribosome begins as separate 50S and 30S subunits. During initiation, the 30S subunit binds to both the mRNA and FMET-ternafmet, forming the 30S initiation complex. The 50S subunit then joins to form the 70S initiation complex. Initiation factors play key roles. IF3 binds to the 30S subunit. IF1 binds to the A site and helps position IF2. And IF2 hydrolyzes GTP to assist with the binding of FMET-ternafmet. Once the large subunit joins, GTP is hydrolyzed and the initiation factors are released, completing the initiation process. In bacterial mRNAs, the Shine-Dalgarno sequence plays a crucial role in initiation. This purine-rich consensus sequence, located just upstream of the August start codon, base pairs with a complementary sequence near the 3' end of the 16S rRNA in the 30S ribosomal subunit. This interaction aligns the start codon with the P site of the ribosome, ensuring that translation begins at the correct site. In eukaryotes, the initiator tRNA is charged with a non-formulated methionine, unlike in bacteria. However, eukaryotes still use two types of tRNAs, an initiator met-tRNA and an elongator met-tRNA. Eukaryotic mRNAs do not have Shine-Dalgarno sequences. Instead, the ribosome locates the August start codon within a specific sequence called the Kozak sequence, typically a call. This discovery, proposed by Marilyn Kozak in 1978, led to the development of the scanning model of translation initiation. Eukaryotic initiation follows 6 key stages. First, EIF2 binds GTP and met-tRNA to form a ternary complex. Second, this complex associates with the 40S ribosomal subunit, aided by other initiation factors. Third, the mRNA is activated by removing secondary structures. Fourth, the mRNA binds to the ribosome complex. Fifth, the ribosome scans the mRNA from 5 to 3, looking for the start codon. Finally, the 60S subunit joins, forming the full 80S initiation complex. At this point, elongation is ready to begin. In eukaryotes, translation is tightly regulated by phosphorylation, particularly of EIF2 alpha, a subunit of EIF2. Normally, EIF2 GDP must be converted back to EIF2 GTP to initiate a new round of translation. However, phosphorylation of EIF2 alpha blocks this exchange by sequestering EIF2B, the GTP exchange factor. 4 protein kinases, each responsive to different cellular stresses, can phosphorylate EIF2 alpha. This mechanism allows cells to modulate protein synthesis in response to environmental cues such as stress, nutrient availability, or viral infection. The elongation stage involves the step-by-step addition of amino acids to the growing polypeptide chain. In bacteria, this process requires elongation factors EF2, EFTs, and EFG. In eukaryotes, their counterparts are EF1A, EF1B, and EF2. These factors help deliver charged tRNAs to the ribosome, ensure proper codon-anticodon pairing, and drive the movement of the ribosome along the mRNA. During elongation, EF2, bound to GTP, delivers an aminoacyl-tRNA to the A site of the ribosome, forming a ternary complex. At this point, the tRNA is in an A/T configuration, where the anticodon is interacting with the mRNA, but the amino acid end is still bound to EF2. This positioning ensures that only correct codon-anticodon matches lead to the next step in peptide bond formation. When the codon-anticodon pairing is correct, EF2 hydrolyzes GTP, triggering a conformational change. This allows the amino acid end of the tRNA to move fully into the peptidyltransferase center of the ribosome in a process known as accommodation. This precise movement is crucial for accurate and efficient peptide bond formation. Once the aminoacyl-tRNA is in the A site and the peptidyl-tRNA is in the P site, the ribosome is primed to form a peptide bond. Remarkably, this reaction does not require ATP or GTP. Instead, the catalytic activity is carried out by the 23S rRNA, classifying the ribosome as a ribozyme. The growing peptide is transferred from the P site tRNA to the amino acid on the A site tRNA, elongating the chain. After peptide bond formation, the EF-TUGP complex is released and must be recharged with GTP before it can function again. This exchange is facilitated by another elongation factor, EFTs, which removes GDP from EF2 and allows GTP to bind. In eukaryotes, this function is performed by EF1B, enabling a continuous supply of active elongation complexes during translation. The hybrid states translocation model explains how tRNAs move through the ribosome. After peptide bond formation, the A site tRNA now holds the growing peptide, while the P site tRNA is deacylated. These tRNAs shift into intermediate hybrid states, A/P and P/E, where they are bound partially to both ribosomal subunits. EF-GGDP then drives the final translocation step, converting these hybrid states into fully translocated positions, returning the ribosome to the classical configuration and preparing it for the next round. In this final step of elongation, EFG bound to GTP catalyzes the translocation of the ribosome. As GTP is hydrolyzed, the peptidyl-tRNA moves from the A site to the P site, while the deacylated tRNA shifts to the E site and exits. The A site is now vacant and ready to accept the next aminoacyl-tRNA. This cyclic mechanism ensures the smooth and rapid elongation of the polypeptide chain. Translation ends when a stop codon, UAA, UAG, or UGA, enters the A site. In bacteria, release factors RF1 and RF2 recognize these codons and facilitate the release of the polypeptide from the P site tRNA. A third factor, RF3, enhances the rate of peptide release. In eukaryotes, ERF1 recognizes all three stop codons, working alongside ERF3 to ensure accurate and efficient termination. Occasionally, a mutant tRNA can suppress a stop codon mutation by inserting an amino acid instead of terminating translation. To summarize, translation proceeds through 4 orchestrated stages. Initiation aligns the ribosome and mRNA. Elongation adds amino acids via repeated codon recognition, peptide bond formation, and translocation. Termination releases the completed polypeptide upon encountering a stop codon. Finally, recycling prepares the ribosome subunits for a new round of protein synthesis. Each step is driven by energy and guided by specialized factors.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
After termination, the ribosome must be disassembled and recycled. In bacteria, this process is assisted by the ribosome release factor, EFGGDP, and initiation factor, IF3. These elements work together to dissociate the ribosome from the mRNA and tRNA, allowing the subunits to be reused. In eukaryotes, the recycling mechanism is not yet fully understood, but the concept remains. Ribosomal components must be reset for another round of translation. The newly synthesized polypeptide begins folding as it exits the ribosome through the exit tunnel, which can only accommodate about 60 amino acids in an alpha helix. Larger folding must occur outside the tunnel. Associated enzymes assist in post-translational processing, such as peptide deformylase, which removes the formyl group, and methionine aminopeptidase, which cleaves the N-terminal methionine in many proteins. These early steps are essential for final protein structure and function. In bacteria, a chaperone known as trigger factor binds to the emerging polypeptide, preventing misfolding and aggregation. In eukaryote, similar chaperones associate with the ribosome to assist in protein folding. Proper folding is vital. Misfolded proteins are associated with diseases such as Alzheimer's, Huntington's, and Parkinson's. These chaperones are part of the cell's quality control system, ensuring proteins achieve their correct 3-dimensional shape. Some proteins carry a signal sequence, a short stretch of amino acids at the N-terminus that determines their cellular destination. Proteins without a signal sequence remain in the cytoplasm or mitochondria, while those destined for secretion, the plasma membrane, or lysosomes, are directed to the rough endoplasmic reticulum RER. In eukaryotes, translation begins on free ribosomes, but when the signal sequence is present, the ribosome is recruited to the OR membrane. The signal recognition particle SRP binds to the signal sequence, temporarily halting translation. The SRP then guides the ribosome to the SRP receptor on the OR membrane, where it docks with the translocon, a protein-conducting channel. Translation resumes, and the growing polypeptide is threaded into the lumen. Inside the the signal peptidase enzyme cleaves the signal sequence, finalizing this stage of protein targeting. Figure 16.55 illustrates the mammalian SRP cycle, showing how the signal sequence, SRP, SRP receptor, and translocon coordinate to direct protein synthesis into the OR. This elegant system ensures that proteins reach their proper cellular locations, a process vital for cell function and intercellular communication. Once inside the ER, proteins undergo further folding, modification, and sorting to fulfill their final roles.